I'm Rachel Evans.
I'm Rachel Summers.
And we did our enzyme project on beta-glucuronidase. So just a brief outline, we're going to go over the function and structure of our enzyme, and we'll go through inhibitors that bind to the active site, and the location of our enzyme on the chromosomal and cellular level, and then we'll look at similar proteins of the enzyme, and then we'll go over the methods and results of the research paper we chose to review. So first we'll go over the function. So beta-glucuronidase, or GUS, functions in the degradation of glucuronic acid containing glycosaminoglycans, or GAGs. So they catalyze the hydrolysis of the GAGs by cleaving at the beta-D glucuronic acid residues at the non-reducing termini of the GAGs. So this is a basic mechanism of the catalysis reaction. So you have the beta-D glucuronides and water as the reactants, and alcohol and D-glucuronate as the products. So now we'll go over the structure of our enzyme. So this is an image of the beta-glucuronidase. It is the hollow enzyme, or the functional form, is a tetramer containing 4 identical subunits composed of alpha helices and beta sheets. And the enzyme is a member of the beta-glycosidase, which is part of the hydrolase class. And each subunit has 3 distinct domains. So it has the TIM barrel domain, the jelly roll domain, and the immunoglobulin constant domain. Oh, and together they function in lysosomal targeting.
Okay, so this is the amino acid sequence of human beta-glucuronidase, which we got from Uniprot. And what we did was we highlighted in red the hydrophobic regions of the actual enzyme itself, so you can just see that. And we also broke it down amino acid by amino acid to show the composition within the enzyme. So you can see that leucine and valine are the most prominent, although it's not much more than the others. But these molecules are both small and have no charge at a pH within the body. But other than that, yeah, there's no other major outliers. And so here is another picture of beta-glucuronidase. You can see here again the subunit, and each color represents one subunit, all being the same. And here in pink is a beta hairpin loop, which is present throughout. And here in blue are actual amino acid residues that are involved in catalysis with beta-glucuronidase. And here in the orange are end-linked oligosaccharide chains. And again, all 3 of these things are present in every subunit throughout the enzyme. And so now we're going to talk about some inhibitors that work on the active site of beta-glucuronidase. And so these inhibitors have been suggested recently to have anti-carcinogenic properties because they actually decrease the amounts of glucuronidated carcinogens that are present in the tissues. And so beta-glucuronidase is a key mediator of drug-induced toxicity in the human GI tract and other tissues within the human body. And some more recent studies have been done showing that beta-glucuronidase inhibitors can be used with chronic drug treatment, and so it reduces the negative side effects that are often seen with drugs such as chemotherapy with cancer treatment. So these are just a couple examples of some inhibitors that work on beta-glucuronidase's active site. And so you can see that all 3 of these have a high presence of ring structures and OH groups, which allow for lots of hydrogen bonding to the active site.
So now we'll talk about the location of our enzyme. So our enzyme is found on chromosome 7 on the long arm indicated here by the yellow line, and the cellular level, it's found in lysosomes, which function in the digestion and removal of waste within the cell, and so that corresponds to the function of our enzyme. So some similar proteins we looked at for our enzyme. So we looked at the human beta-glucuronidase compared to the bacterial. So the human here is shown in red, and the green is the bacterial, and the images are superimposed, so you can see the similarities and some differences between the two structures. And you can see all the alpha helices and beta sheets, kind of how they align. So zooming in, this is the lysosomal targeting loop, and you can see in the human there's some extra twists in the alpha helice compared to the bacterial form of the enzyme. And then the other image is of the active site, and you can see here in bacterial has an extra 17 amino acid residues, and this plays a role in the inhibition and catalysis that allows for it to be a therapeutic target against the bacterial form of the beta-glucuronidase. And so this is another image comparing the human form of the enzyme to E. coli and mouse. And you can see in the blue alpha helices and the green beta sheets. And you can see here with the human and the mouse lacking the additional 17 amino acid residues found in the bacterial form. And the green here highlights the active site residues, and the pink highlight the glycosylation sites. And the dark gray areas represents the three species having the same amino acid residue structures.
So the study that we decided to look more in depth on was called the Interindividual Variability in Expression and Activity of Human Beta-Glucuronidase in Liver and Kidney and its Consequences for Drug Metabolism. And so the purpose of this study was to basically investigate the variability in expression and function of the beta-glucuronidase enzyme within the human kidney and liver tissue. And so variability in expression of this enzyme can be a confounding factor for interindividual variability in drug disposition and in the setting of the accumulating of these glucuronides. And so glucuronides of drugs actually accumulate during this chronic therapy, like I mentioned earlier, with chemotherapy and other treatments like that. So to go through the methods that they used for this study, they first began with some tissue samples. They got human liver and kidney tissue samples as surgical waste following partial hepatectomies or nephrectomies. And they immediately stored these and froze them in the -80 freezer. They performed an HPLC analysis, which is a high-performance liquid chromatography. This is basically just a better form of a chromatography, better than column chromatography or thin layer chromatography like we did in organic chemistry. But what makes it better is that it injects air into the column, so it just speeds up the mechanism a little bit faster. And so for beta-glucuronidase, the substrates that were used were MUG and DOX, and they found that their retention times were 4.5 and 7.3 minutes, basically just showing that DOX had a higher affinity for the solid phase within the column. They also did a Western blot analysis, which we recently talked about in class as being more of a qualitative analysis to determine the presence of the enzyme within these tissues. And so to do this, they took 50 micrograms of both tissues from liver and kidney and subjected them to a STS gel with a anti-beta-glucuronidase antibody. So for the results that they found, they resulted in specific beta-glucuronidase activity, which basically is just a measure of the expression of this enzyme within the tissues. And so from these charts here, you can see that the liver has a higher presence of beta-glucuronidase than the kidney, because the kidney has its curve is shifted more towards the left versus the liver, which has a more of a right shifted curve showing the higher presence of the enzyme. And so the Western blot, they found that in both the liver and the kidney, the enzyme was stopped at 64 kilodaltons, which makes sense because this is the molecular weight of beta-glucuronidase, but basically, again, just showing that it is present in both of the tissues. And so they also quantified the enzyme kinetics of beta-glucuronidase with using two different substrates, again, the MUG and the DOX. And so you can see here from the MUG that it has a hyperbolic Michaelis-Menten plot, which we just talked about in class, but you can see that the liver actually has a higher Vmax than the kidney. And then over here you can see that DOX actually has a sigmoidal velocity curve, but again, the liver tissue has a higher Vmax than the kidney. And so being that with both substrates, the liver had a higher Vmax, this could be due to just the higher presence of the beta-glucuronidase enzyme within this tissue.
So in conclusion, beta-glucuronidase is a lysosomal hydrolase that functions in the hydrolysis of beta-D-glucuronides into glucuronic acid and aglycone. And the enzyme is a tetramer that has 4 identical subunits containing alpha helices and beta sheets.
And again, the study that we focused on found that the beta-glucuronidase enzyme is present in both the kidney and liver tissues. However, other studies have been done showing that beta-glucuronidase has a high presence in cancerous tissues and other tissues throughout the human GI tract and so on. And more studies have been done showing that beta-glucuronidase inhibitors can actually reduce the negative side effects of chronic drug treatment such as cancer chemotherapy. And so these are the references that we used, and here specifically are the bioinformatics sites that we used. We just wanted to thank you for watching.